Good afternoon, everyone. I would like to present to you today some technical aspects of the detection of Plasmodium falciparum and Plasmodium vivax gametocytes. And I would like to discuss with you our findings on the infection dynamics in a cohort study in Papua New Guinea. Before we started off with tracing gametocytes, we were not quite sure how to sample in this huge cohort study. So the entire study then, I will report, was sampled on filter papers, and then that was stored in Trisol to keep the RNA. We also then did a parallel study to assess another method, which is RNA Protect, which we found was the much better method because the yields of RNA were better and the handling was much better too. So that was, we discovered that a little bit too late. So this also explains a bit the limited sensitivity, in particular for Plasmodium vivax gametocytes, what we found, which Ivo has explained already, because we have a 10-fold lower vivax asexual parasitemia, and this also translates, of course, in a lower vivax gametocytemia. So RNA is the quality of the RNA is absolutely essential for all these molecular tools. So one has to take great care and optimize sampling and the fieldwork, of course. So we have also optimized or investigated the use of molecular markers for Plasmodium falciparum. We have used here the standard protocol would be PVS 25 and PFS 25 for vivax and falciparum. But we thought it would be ideal to have a marker with an intron, so we could avoid the DNase treatment, because we know DNase treatment is detrimental to RNA too. So we could assess first the detection limit of other markers, but we also could assess the effect of DNase treatment. So we have compared with this marker DMC1, which has an intron. We have treated that marker with DNase, and we have done it straight on RNA, because that we can do when the amplicon is spanning the intron. So that is a 50-fold loss when we are forced to do a DNase treatment. However, when we use the highly expressed PFS25 transcript, this is still more sensitive. So, I mean, we didn't see a reason to change from PFS25. We have also assessed another marker recently described as specific for gametocytes by the group of Matthias Marti, and we also have seen the same thing, a 50-fold loss by DNase treatment. So the conclusion of that is we were fine with PFS25. So that remains our marker despite the loss and despite the fact that we have to do an additional quantitative PCR to detect for remaining DNA. So the limiting factor here is probably the blood volume. If we could increase the blood volume, we would have a lot more sensitivity. So we have sampled 50 microliters, and in these 50 microliters we have enough sensitivity to detect one single gametocyte. So if we would sample 100 microliters or 150, of course, we would increase the sensitivity. So this is for fieldwork, this is something to consider. Now I'm coming to the cohort study, which was conducted 2008 to 2009 in PNG, and that was a two-armed trial. And the reason for this cohort was to study the effect of primaquine treatment on the incidence of disease, the molecular force of infection, and of course gametocide prevalence. So that involved the 4-week treatment of primaquine plus chloroquine treatment and coartem. In the primaquine group, in the placebo group, it was only chloroquine and AL. So there were 500 children enrolled in that cohort study over 8 months, and it was they were 5 to 10 years of age. So there was a dense sampling, but possibly not dense enough, as we know now. So that was the baseline sample, then this 4 weeks treatment, and then 2 weekly bleeds, and after this 1 monthly bleeds. In parallel, active case detection and passive case detection, so we have sampled all cases as well. So here we see the effect of primaquine on gametocides, on Plasmodium vivax gametocides. This is a time to first Plasmodium vivax gametocide infection in the 2 arms. The black is the primaquine arm and the placebo arm in red. There is a major difference. The primaquine treatment is associated with a 70% reduction in the hazard of becoming positive for Plasmodium vivax gametocides. So this is a very strong effect. And what's important also to see is that initial three months, there the effect is strongest. That, of course, can be explained by the new infections also then contributing to relapses, and the effect of the primaquine treatment would wear off here. So we also were interested to see whether there was a delay in parasite infections whether gametocides appeared a little bit delayed or together with the Plasmodium vivax infections. So this graph shows us again that the black curves are the ones you have seen before. That's the primaquine group and the big difference to the placebo group. And here in red we have the simultaneous detection of asexual and gametocides, and in gray the delay in the detection of gametocides. There is no difference here in the placebo group and no difference in the primaquine group. If we compare this to Plasmodium falciparum, which is sympatric and transmitted by the same mosquitoes in the study area, we see there is certainly not this major effect of the primaquine treatment, a little bit of an effect. In the placebo group, we also don't see a major increase in the delay in the time to gametocide detection. But in the primaquine group, we see a significant effect. So there the onset of gametocytemia seems to be delayed. We further were interested in studying how much relapses contribute to gametocytemia compared to new infections. So this could be assessed by comparing the 2 study arms. But we looked here only at the Plasmodium vivax positive samples in the 2 arms, and obviously these were much less in the primaquine treated arm. So here we see the percentage of gametocide positives in these samples, and the confidence intervals overlap. There is basically no major difference over the entire study. So we concluded from this finding that relapses are the same as new infections in their contribution to gametocides in these children. So we can conclude this. Children with primagreen had 4 times children without primagreen treatment were 4 times more often positive for gametocytes. So the primagreen has a major effect here. We have seen that relapses equally produce gametocytes, and that could suggest that relapses, because of this massive effect here, relapses are likely the primary source of Plasmodium vivax transmission. I think this could be a strong evidence base for using primagreen as a transmission blocker in the context of elimination programs. Second, I would like to come to another aspect of that study. We would like to continue and genotype the individual gametocyte clones. So we were looking for markers for highly sensitive and gametocyte-specific molecular markers, if possible those with an intron, so we can avoid the DNase treatment. And we wanted to use length polymorphic markers with a large number of alleles. We use capillary electrophoresis to distinguish the individual gametocyte clones. So we have assessed a couple of known markers. These are the famous one, PFG377 and PFS230. But also sort of new markers, which we expected to be length polymorphic because they have intragenic repeats. So some markers had introns, others didn't have introns. So the sensitivity is the crucial parameter here, and we found that the sensitivity of PFS 230 is the best one, so this is the ideal marker. We could improve this marker a little bit by including 2 repetitive regions, and we also assessed PFG377. That is a little bit less sensitive because the amplicon size is much larger. So the diversity of that marker, PFS 230, including 2 regions as well, was assessed on DNA level in PNG. So we had in 95 samples, we found 17 genotypes. This is a quite high resolution for a genotyping marker. So we were quite pleased with this. Then coming to the comparison of DNA and RNA. So we directly compared the genotypes found in DNA and RNA in 42 samples, and this is a poster which was presented today at the poster session. What we see here is in white, we see the genotypes which are only observed in DNA. That is what you expect, because maybe not all clones produce gametocytes. So we have a lot of clones have in parallel on RNA level and DNA level detected, were positive for both. So this would be the clones who actually produce gametocytes, but to our surprise, we also found a lot which are only detected on RNA levels. So many individuals had a clone which was not seen on DNA level. And this could maybe be a minority clone which is suppressed during PCR on DNA level, but in the mosquito it might be more prevalent, more gametocytes, and then suddenly it appears. So that was a bit a surprise, and it means we possibly have to look at this in a longitudinal way. So there are issues with detectability of individual gametocyte clones. There is imperfect detection at both levels, in DNA level and RNA level, that has been described before. As I've said, larger volumes for sampling RNA could help here. We have had a smaller volume. But it also highlights again the expression levels are utterly important for gametocyte-specific markers. The higher, of course, the better. And the preservation of DNA and RNA is also important because these amplicons of polymorphic genes are really long to get enough diversity, so the RNA must remain well intact. We thought that a longitudinal approach will resolve this. That is our next aim, because it's possible that we see a gametocyte which is sequestered at an earlier time point, maybe at the time point 2 weeks later. So this would be our continuous work to study the infection dynamics of individual parasite clones. So I would like to acknowledge my students who have developed these essays in Basel, and our close collaboration with Ivo Müller and the PNG IMR led by Leanne Robinson, and funding comes from different organizations. I need to highlight the TransEpi Consortium because this is a TransEpi session here. Thank you very much. And lastly, I would have a little advertisement for a congress in my hometown Basel in Switzerland. Maybe you are interested to come there next year. Thank you very much.
Thanks, Ingrid. We'll take just a couple of questions.
Ingrid, that was great as usual. Maybe I missed something, but in your comparative trial for falciparum, there was a considerable delay in primacrine treated kids for gametocyte carriage, right? That was several weeks. How would you explain that?
I cannot explain that.
But the impact is really heavy, right?
Yes.
If it would just delay carriage, but not remove it only slightly. So it's a major information for falciparum.
Yes, the onset of gametocytemia was substantially delayed. Yes. Mm-hmm.
Thank you, Ingrid. Very nice. What proportion, I might have missed it, so point of clarification.
Yes.
What proportion of your PV gametocytes were present without an asexual parasitemia?
What proportion? I mean,
You measured PV gametocytes. What proportion? So what?
Yes. You mean in the cohort study with the two arms?
Yes.
The way how we genotyped was that, as Ivo has shown the diagram, we only looked for gametocytes in the DNA positive samples. So we first screened for Plasmodium vivax on DNA level, and then we only checked there for gametocytes. We didn't do the negative samples.
Because you could argue...
It's just too many samples.
Okay, but you could argue that actually, if the dynamics are completely different, and that if you've got asexuals present, you've got gametocytes present, and as we've shown, that's even at subpatent levels that we're not detecting, in which case, do you need to go to the RNA, or why don't you just look at the asexual carriage, which might actually be easier to do?
In the mosquito, we could actually do... You mean for genotyping?
In the patient?
Yes. I didn't really get the question.
Sorry. In your carriage of gametocytemia, which you've shown has been reduced by primaquine, which is a great observation, and showing that you're going to decrease the transmission potential of vivax by primaquine use, radical cure, which is fantastic.
Yes.
Do you need to, as a marker of that, do you need to actually be looking for PV gametocytes, or can you?
No,
you don't need to.
No, not
okay,
at all.
I missed the point. Thank you.
No, not at all. No, I mean, of course, you can look at that on for molecular force of infection, as we have done it, or by microscopy, or by incidence, by whatever. You see the effect. But in this particular session, I wanted to just present the transmission, the gametocyte data.
A very important point.
Yes, it is an important point, but Ivo has touched upon the other data already in the session on Monday.
Okay, I think we'll have to move on. Thanks, Ingrid.